Welcome back. This is Chapter 5, Part 2. We're moving on from chromosomes to the chromosome theory of heredity. Now, today we know that genes sort of live on chromosomes, that chromosomes contain DNA, which contains the information for genes. But this wasn't always known. Now, scientists knew about genes and they knew about chromosomes, but they needed proof that they had some link to each other. The first scientist to perform experiments in an attempt to answer this question of whether chromosomes are composed of genes was Thomas Hunt Morgan. Now, to answer this question experimentally, he needed three things. First, he needed to find a gene that was unambiguously or definitively associated with a chromosome. Secondly, that chromosome must be distinguishable from the other, so he had to be able to follow it from parent to offspring. And third, he needed to be able to follow the pattern of the gene transmission to the offspring. So he had to be able to follow how the gene itself was being transmitted from parent to offspring. And he had to show that that was consistent with the behavior of the chromosomes during reproduction. So he had to show that the pattern of the gene being transmitted from parent to offspring followed the pattern of the chromosome as it was transmitted from parent to offspring. So the model that he used for this was Drosophila, the fruit fly, and he looked at eye color, the eye color trait. Now, Drosophila is fairly easy to work with. They only have 4 chromosomes, which are easily visible. And he knew that eye color was actually associated with the X chromosome. Now, what's important about this particular experimental design was that he could easily follow the pattern of eye color in males versus females. I'll go back to that in a second. And because they are recessive, if they were associated with the X chromosome, they would always show up in the males. So essentially, Morgan's experiments, as a sort of a preview here, on this sex-linked trait actually provided the first evidence that the behavior of chromosomes during meiosis is the actual basis, the actual foundation for what Mendel described as in his principles of segregation and principles of independent assortment. All right, so let's look at this in a little more closely. Okay, so again, the eye color gene is present on the X chromosome, but not on the Y chromosome, keeping in mind that the X and Y chromosomes are different in terms of the genes that they contain. Now, red eyes are dominant, white eyes are recessive. So the first thing he did was cross a homozygous male with red eyes with a hemizygous male with white eyes. Now, hemizygous refers to the fact that males only have one X chromosome. Because of that, and because these sequences are different than what we see on the Y chromosome, males can't be homozygous or heterozygous for a trait on the X chromosome because they only have one. So we refer to them as hemizygous. So that's important here. And this is why, as I mentioned earlier, recessive traits that are on the X chromosome will show up in hemizygous males because there's only one X chromosome. Okay, now we can easily predict what's going to happen in this cross because what we want to do is observe the eye color allele and see if it behaves as chromosomes do. Now, as I said, it's easy to kind of predict what we're going to get based on the gender simply by looking at this cross here. So this female is going to give a red-eyed allele to all of her offspring, but the males are going to give an X white-eyed allele and a Y chromosome to his offspring. So this is pretty straightforward. What you're going to end up with is red-eyed females and red-eyed males. And when you cross these F1s together, you're going to get 4 different populations, so to speak. Homozygous red-eyed females, heterozygous red-eyed females, red-eyed males, hemizygous, and white-eyed males, hemizygous. Now, this is easy to see using this particular experimental design that Morgan chose, because we can say, well, we can follow the transmission of this X chromosome from this parent to this offspring to these offspring. Okay, it's easy to follow that along because we can simply score whether they're male or female and whether or not they have red or white eyes. Let's look at another example and make this a little clearer as well. So again, we have our red eyes that are dominant, white eyes are recessive, and we're going to cross a heterozygous F1 female with a hemizygous male that has white eyes. And again, we can predict if the X chromosome actually carries that eye color allele. We can predict that we're going to get these heterozygous red-eyed females. We can predict that we'll also get white-eyed females. We can predict that we're going to get red-eyed males and white-eyed males. And we can predict what ratios we're going to see these in just by using classic Mendelian monohybrid crosses and what we would predict in the offspring. And that's what Morgan did, and he predicted what he would see if these two things were linked. That is, if the allele was actually carried on the X chromosome. Next, he took a white-eyed female, homozygous, crossed it with a red-eyed male, and again, by predicting what he would see in the offspring, whether the X chromosome is going to be following essentially the transmission of that allele, we can see that there is actually a link between where the X chromosome goes and where we see that white-eyed trait. So these experiments led Morgan to his chromosome theory of heredity, which is really just as important as Mendel's principles. Now, what this theory states is that by correlating the inheritance of a trait, in this case eye color, with the transmission of the X chromosome, Morgan was able to prove that the gene for eye color was actually present on the X chromosome. And he next surmised that because of this, all genes must be located actually on chromosomes, and this was the basis, or this basically led to his famous chromosome theory of heredity. Now, there was actually a problem with this theory. Sometimes there are exceptions to this rule. Sometimes, for example, he saw red-eyed males after a mating between a homozygous white-eyed female and a red-eyed male. Now, normally what you would expect to see is in a male would be the white-eyed allele and a Y chromosome, which should give you a white-eyed male, and this is essentially what you should have all the time. But sometimes he saw this particular case here. How could that be? How can he explain these exceptions and still stay true to this chromosome theory of heredity? Well, in fact, there was an explanation, and that is sometimes chromosomes behave abnormally during meiosis. This behavior is called non-disjunction, and this is what happens when the chromosomes do not segregate properly during the formation of the gametes. So, for example, if this happened in the egg or in the ovary of this female, you would end up with some normal eggs that carried what you would expect, this white-eyed allele, but also some non-disjunction eggs, where some eggs ended up with two X chromosomes and some ended up with none. And that does happen. So by looking at, by recognizing that these abnormalities can happen, Morgan was able to make these conclusions that, well, the X chromosome is actually needed for viability, that's because the males that only received a Y chromosome don't survive. However, the Y chromosome is also needed for fertility, but not sex determination, because we could see that we could have males here. Okay, so these flies retained their maleness, but they were sterile, they were infertile. All right, so let's see if we can merge or marry Morgan's chromosome theory of heredity with Mendel's principle of segregation. Now again, Mendel devised this principle of segregation. He knew that the alleles segregate during the formation of the gametes during meiosis. But this chromosome theory of heredity kind of proves what why that happens, or essentially what the basis is. And what I mean by that is if you have a cell here, okay, and here we have the maternally inherited chromosome and the paternally inherited chromosome, and then this DNA is going to replicate during meiosis, as we know what happens. But then these chromosomes are going to physically separate. Okay, you're going to get one chromosome going to one end of the cell, one chromosome going to the other end, so that they end up in different daughter cells. And so essentially what's being said here is that this physical separation of the chromosomes during meiosis is the basis, or essentially the chromosome theory of heredity explains that the principle of segregation is based on this physical separation of the chromosomes. Now we can also use the chromosome theory of heredity to explain Mendel's principle of independent assortment. Now again, we're going to look at two different traits. These traits that are going to segregate independently. So we have an AA-BB. So here's the recessive A, dominant B, recessive B on different chromosomes, keeping in mind these are the maternally inherited chromosomes, these are the paternally inherited ones, and they have already gone through DNA replication. And what's going to happen is, again, the chromosomes are going to physically segregate during meiosis. And because these alleles are going to segregate independently, in these final gametes here, you're either going to have gametes that contain two dominant or two recessive, or if they line up differently on the metaphase spindle, you could end up with a big A little b and a little a big B. And essentially, because we know that these alleles are carried on chromosomes, okay, as a result of the chromosome theory of heredity, then we know that by segregating these chromosomes, that is the basis that explains Mendel's principle of independent assortment. Now this concludes part 2 of chapter 5. Please join me again for part 3.